bit better? Because I'm really proud of some of this staining, so I want you to be able to see it. Okay, so for those of you who haven't met me, my name is Megan Simpkinson, and today I'm going to be presenting my thesis, which is novel improvements to the differentiation of human iPS and ES cells for the derivation of pancreatic beta cells. And I'd first like to introduce my committee. Professor Alberto Hayek is the chair, and Cornelius Murr is the co-chair, and Elvira Tour is on the committee, and they have graciously agreed to help me with my thesis, and we'll be picking my brain in a few minutes, I'm sure. All right, so the staining that you see in the background, just to introduce it because it's pretty and you're probably wondering what it is these are cells that have, they're iPS cells that were taken to the end of the differentiation protocol that I'm going to be introducing to you in a few minutes. And I stained for PDX in red and insulin in green. Actually, I think this was C-peptide staining. So in green, and just wanted to put that in the background, but you'll hear more about that in a moment. Okay, so I'd first like to start by talking about the reason that we're all here, which is looking for a cure for type 1 diabetes mellitus. It is caused by destruction of the pancreatic beta cells due to autoimmunity, and it is a lifelong illness that is treatable only through injections of insulin or insulin pump therapy. The major downside is that a lack of tight control leads to dangerous hypoglycemic incidents and long-term complications such as diabetic retinopathy, neuropathy, and kidney disease. And it affects 3 million people in the United States. Currently, the only cure for diabetes comes in the form of islets from cadavers. However, these have a couple of negative side effects. The main issue is that anyone who receives these cells has to be under severe immune suppression for the rest of their life, and there's all kinds of bad side effects to that. And they're also a limited resource, and there are not enough islets available to suffice for all of the diabetics that could benefit from them. So researchers have turned to embryonic stem cells and induced pluripotent stem cells as a potential new resource for beta cells. Both ES and iPS cells can be differentiated to become any type of cell and are a renewable resource. Insulin-producing cells have been generated from both embryonic stem cells and iPSCs at this point, but we still have a long way to go before these cells can be introduced into a clinical setting. All right, so I'd like to first talk about the generation of embryonic stem cells and iPSCs to give you a little bit of background. So embryonic stem cells were first isolated and cultured in 1998 by Thompson and colleagues. 5 days after fertilization, blastocyst, which is over here, has formed, and the inner cell mass, which we'll see another picture in a moment. This is a blastocyst, which consists of about 200 cells. Beautiful picture. And this is basically the blastocyst opened either through chemical or mechanical means to reveal the inner cell mass. And it's from this inner cell mass that the stem cells are isolated and can be cultured. All right, so next, iPS cells. iPS cells can be generated from a number of different tissues. In these studies, our cells came from type 1 diabetic mellitus patients' fibroblasts. And the way that they were generated is you start by taking a skin punch from the arm of a diabetic patient, in this case, a male diabetic.
It's my eyes, so
it's not very strong. Does anybody else have a stronger laser pointer with them? No? Okay, well I can use my mouse. Okay. Okay, well I'll try and make do until then by just pointing. So you can see that there was a skin punch taken here, and the fibroblasts are then isolated from that and reprogrammed using pluripotency transcription factors. And you can see that after reprogramming, they express several different stem cell markers. They will stand for alkaline phosphatase, and they also stand for OCT4, TRA160, NANON, TRA180, SOX2, and SSEA4, showing that at least some of the cells have been able to be reprogrammed to an induced pluripotent stem cell fate. All right, and the benefits of these cells is that they are patient-specific, which means that they can be taken from a patient for a patient. We can take the cells from this diabetic, generate iPS cells, and hopefully someday be able to then generate beta cells that can be put back into that patient to cure his or her diabetes. And there are some hurdles, however, that need to be overcome. We do see that there have been some mutations that are introduced with reprogramming in some cases, and we need to be able to address those concerns before these cells can be applied to clinical applications. All right, so next I want to talk about the current differentiation protocols. The most successful published protocols to generate beta cells from stem cells mimic the normal in vivo pancreatic development. And we use a modified version of the Diamore et al. 2006 protocol. Thank you. And this protocol attempts to mimic in vivo pancreatic beta cell development by taking the cell through 5 different stages. Begin over here with the embryonic stem cell or iPS cell, and you, by applying certain growth factors and signaling molecules, we go through a definitive endodermis stage, then to a primitive gut tube, next to posterior foregut, then pancreatic progenitors, and finally to hormone-expressing endocrine cells. And while this protocol has been able to generate insulin-producing cells and has even had some success in vivo, there are some points of potential improvement, and this is kind of where we're going to start getting into what I wanted to address. First of all, induced pluripotent stem cell differentiation is not as efficient as embryonic stem cell differentiation. In fact, this was the first time that we were applying the Diamore protocol to iPS cells. And the development of pancreatic endocrine progenitors is not yet optimized. Also, NGN3 and PDX1 transcription factors, I just want to introduce those really quick, are expressed in the cells that will become pancreatic endocrine cells, and you'll see later that we're going to be looking at those as 2 very important transcription factors for differentiation of these cells. As a result, the final yield of insulin-producing cells is on average only a small percentage of the cells generated. This brings me to the questions that I am addressing in my thesis. First, can we improve induced pluripotent stem cell differentiation by optimizing the Diamore protocol for these cells? Also, can we improve differentiation of both embryonic stem cells and iPSCs by finding new small molecules to influence development of endocrine progenitors? And finally, will induced pluripotent stem cell derived cells be able to generate insulin and respond to glucose challenge after transplantation into immune suppressed mice? This has so far not been accomplished. No one has been able to transplant iPS cells successfully. Okay. So, starting with the first question, can we improve iPSC differentiation by optimizing the protocol? We attempted to optimize the length of time that the iPS cells were kept in the 2nd and 3rd stages, and we looked at this using qPCR that you'll see in a moment, as well as staining at the end of the protocol, and we found that shortening the 2nd stage to 2 days and extending the 3rd stage of culture to 4 days yielded higher expression of NGN3, PDX1, and the eventual endocrine cells. First, we can see higher levels of expression of NGN3 throughout the different stages of development, with a very high peak here during stage 4. And as I mentioned before, these NGN3 cells are the endocrine progenitors. So it's important to have high transient expression of that transcription factor to be able to generate hormone-producing cells. And we also see that PDX1, which is co-expressed in beta cells, is peaked under these conditions, with stage 2 being 2 days and stage 3 being 4 days under the same conditions. So this is confusing to anyone because there's a lot of timing going on here. So at stage 2, we have shortened by a day to 2 days. Stage 3, we lengthened to 4 days for the optimal conditions here. And we took samples at the end of stage 3, stage 4, and stage 5 to be able to evaluate the expression of these transcription factors. All right, and under the same conditions, insulin, glucagon, and somatostatin were found to be at higher levels. So this is as expected. We had higher NGN3 expression, so we expected to find higher hormone expression. And so that's it for the timing experiments that we did. Except there's also this immune fluorescence staining, which was beautiful. Here we have, this is at the end of the protocol, so the end of stage 5, when we have hormone-positive cells. And we see C-peptide in green and PDX1 in red. And this is, you can see that there's some co-staining, which you would expect to see in a beta cell. You would expect it to see both PDX1 expression and C-peptide expression. However, you can also see that there are a few cells that are expressing PDX1 but not C-peptide, so it's possible that by keeping these cells in culture, we may see further upregulation, that these PDX1 positive cells could be as of yet immature. So moving on to the second question that I wanted to address, which was, can we improve the differentiation of both embryonic stem cells and induced pluripotent stem cells by finding a small molecule inhibitor to influence the development of endocrine progenitors? So we looked at PP2, which is a Src family kinase inhibitor, because it has been shown to influence the differentiation of other cell types. We decided to test its effects on embryonic stem cells and iPS cells during differentiation. And the addition of Src family kinase inhibitor PP2 induced higher levels of NGN3 expression, as can be seen here by the mRNA expression. And this was at optimized concentrations during addition for the 48 hours at the beginning of stage 3. We were able to induce higher NGN3 expression in Cytera 49, Cytera 203, which are both, and H9, which are all embryonic stem cells, stem cell lines, and also in induced pluripotent stem cells. In turn, we saw protein expression levels were also higher in these cells, and this was confirmed through immunofluorescence staining for NGN3. In PP2 treated cultures, you can see that there's much higher levels of NGN3 in green than in DMSO controls, and this was further confirmed by FACS sorting. We saw a 6% increase in NGN3 positive cells with the addition of PP2. And as expected, this also increased the expression of insulin, somatostatin, and glucagon cells. Here we have a time course that shows the transient expression of NGN3, which was higher with the addition of PP2, correlates with the induction of insulin towards the later stages of differentiation. You can also see that glucagon and somatostatin expression was higher in PP2 treated cells. And this also was confirmed by looking at FACS. We had a 16% increase in PP2 treated cultures of C-peptide expressing cells, and also a 15% increase in glucagon. And over here, we can see staining in ES cell line Cytera 49 for both insulin in red and glucagon in green. This is in the control DMSO. And in PP2 treated, you can see there are a much higher level of expression there. These are both PP2 treated cultures of the IPS line at 38H1. And you can see we get higher PDX1 and insulin expression in both of these. All right, so with all of this in vivo or in vitro data, we wanted to see whether or not we could transplant the iPSCs into mice and be able to eventually cure diabetes, or at least be able to get insulin release with the glucose challenge or secretagogue stimulation. So we transplanted this induced pluripotent stem cell clone CHIP2, which is derived from the 38H1 cell line. And before we transplanted the cells, we tested them to see if they could differentiate, as well as other cell lines that we had tried, and found that they could differentiate to express insulin, as well as a little bit of somatostatin expression at the end of stage 5. And here at the end of stage 3, we saw beautiful staining for NGN3, indicating that a lot of our cells were on their way to becoming endocrine lineage. And in the cells that we actually transplanted, we checked to see whether or not NGN3 and PDX1 expression levels had been induced at the end of stage 3, and they had. We then wanted to look to see if we had any pluripotency transcription factors being expressed in these cells before transplantation, and we found that compared with day 0 controls, we had much lower expression of NANOG and OCT4. All right. However, after transplantation, up to 4 months later, we saw no C-peptide release. And this indicates that either the progenitor cells did not survive transplant or did not continue to differentiate into endocrine cells in vivo. We also saw 50% of the mice developing teratomas, which you can see here. This is a normal mouse kidney, and this is the teratoma that grew at the site where we transplanted the cells underneath the kidney. Upon doing histology of the teratomas, it was revealed that they contain many different tissues, including brain, intestinal tract, respiratory tract, skin, a number of different tissues, showing that they indeed are teratomas. And there was even one that had a fibrosarcoma.
Very naive question. So, could you please describe in a little bit more detail the process of the transfer? So
The transplant?
The transplant. So
Okay.
you basically differentiate the cells
Mhm.
until stage 5 and then transplant them.
Yeah. So those were actually I didn't ever get to perform a transplant, but the process is that we take them to the end of stage 5, and then we trypsinize the cells to bring them up off of the plate and kind of aggregate them and take them from that state and put them into the mice underneath, in this case, the kidney.
But not in the pancreas.
No, not in the pancreas. And I think that's pretty much just because this is more accessible, and we are able to like, the cells there can still produce insulin and glucagon as they would if they were in the pancreas, but this is just a simpler way to do it.
So they show other cells that you show to be able to.
Yes, yeah, there are some studies where ES cells have been transplanted underneath the kidney capsule and been able to continue to differentiate, secrete various hormones.
Historically, of course, pancreas is very stable, so we would like to do replacement cells in the pancreas. In humans, the pancreas is a very nasty gland because if you put cells, needles, or take a shunt, they tend to make fistulas. And since the pancreas produces all the digestive enzymes to very easy to get pancreatitis. So modernism experimentally always went to other places like kidney, because you can see the transplant, because you excise it for distillation examination is very easy. Matter of fact, in humans today, they treat their diabetes with islet transplant. It's the infusion of the cells into the portal vein, so it's like a clot into the liver. And it looks like it's functional, but of course not like pancreas.
Yeah, because of course, the worry is that maybe the specific environment inside the pancreas can promote the correct differentiation of these cells.
Absolutely, yeah. We do have a program to do that, to put cells into the pancreas to see local factors influence differentiation.
Okay, so these teratomas indicate that pluripotent cells were transplanted along with the differentiated cells. So, for the conclusions drawn from these studies, basically altering the timing of the second and third stages of the DMR protocol improved the differentiation of induced pluripotent stem cells, indicating that, as is previously known, the timing of these different stages is very crucial for optimal development. Also, addition of the Src family kinase inhibitor PP2 improved the generation of NGN3 expressing cells, and in turn the number of endocrine cells at the end of differentiation. However, transplantation of induced pluripotent stem cells was not successful. No C-peptide was found up to 4 months later, and 50% of the mice had teratomas at the transplant site.
Just another question about the transplantation experiments. So have you tried transplanting the cells that were differentiated according to the protocol,
Without the alterations?
changes?
Yes, we have. And from Cytera 49, which is an embryonic stem cell line, we took them through the normal DMR differentiation protocol, but we get teratomas in a large percentage of the mice with that protocol as well. And Viacyte, the company that actually provided the Cytera 49 cells, they have shown previous studies where they've been able to transplant another cell line 203 as well as 49 and get lower levels of teratoma formation and be able to even get insulin secretion from those cells once they've transplanted them, but we haven't been able to replicate that so far.
It's a really complicated issue because there is not a single report in the literature of anybody that has replicated this spike from the and I was a consultant and so they have come out with new ways of trying to alleviate the problem of teratoma formation. And we are waiting to see if we can replicate that protocol to see if we get more successful.
Yeah, so that kind of brings me to my next conclusion is that the problem of teratoma formation and successful transplantation of stem cell-derived cells has to be addressed. This is the biggest issue facing this research right now, I feel. And at the same time, while our in vivo studies are not yet perfected, we were able to improve the number of insulin-producing cells in vitro. And these studies have advanced the knowledge of induced pluripotent stem cell and embryonic stem cell differentiation, and have brought diabetics a little closer to replacement beta cells. Some future directions that we need to address and would like to start looking at is obviously the in vivo development. We'd like to look at finding ways to reduce the number of pluripotent cells that are transplanted along with the differentiated cells. And we would like to do so by selecting the cells using cell surface markers and utilizing zinc finger nuclease technology prior to transplant to be able to specifically target cells that are expressing, for instance, PDX1 or other transcription factors of interest, and only transplant those cells into the mice. We're also looking for other small molecules to introduce to the differentiation to continue to improve the in vitro protocol for endocrine and beta cell specification. Okay, and I'd like to just acknowledge a few people. First of all, Alberto Hayek for taking me into his lab and allowing me to do research research here over the last year and a half. I have learned more here than I learned in many of my classes, and probably the most I've learned in a short amount of time ever. And I'd also like to thank Eva Africanova. She has been my guide and mentor throughout my time here, and all of this work would not have been possible without her. And I'd also like to thank Lola. She helped me with all the staying that you see here. She's definitely answered many of my questions regarding that work and many other things, general advice. And I'd like to thank the other members of the Hayek Lab, everybody that I work with on a day-to-day basis. You've all been my teachers at some point, and I really have appreciated all of your help and your lunchtime conversation. And I'd also like to thank Anthony Montgomery. I was able to publish be an author on a paper very early in my scientific career because he let me help out with those studies. And I'd like to thank Myra, even though she's not here right now because she's another author on that paper, and she helped me along the way as well. And then I'd like to acknowledge Yang Xu. All of the iPS cells that we worked with were reprogrammed in his lab, and that's where we get them from. I'd also like to thank Cornelius Murr and Elvira Tour for agreeing to be on my thesis committee and for giving me their feedback throughout this process. And then, of course, my friends and family. My parents are back there. And they've been a constant source of strength and support throughout this whole process. So, thank you to everyone. Our funding sources on these projects were JDRF, CERN, and Hubble. Thank you. And I guess I'll take questions. Yeah, bring the lights. Okay, so I kind of have to take questions from my committee first, so.
Can you elaborate on the zinc nuclease experiment that you were
Yeah,
planning?
so zinc finger nucleases are a technology to be able to go in and specifically you can target specific sites in many different genes. And you go in with this zinc finger nuclease that creates a double strand break and can introduce genes or introduce reporter genes so that you then can select for those cells later on. And it's very highly specific, unlike retroviral transduction or other types of ways of introducing genes into a cell. You can introduce into a very specific site, so you can control where that where the new sequence is integrating.
But why would you want to do that?
Well, if you, for instance, wanted to target cells that, if you wanted to generate cells that could be identified by their, for instance, PDX1 expression, you could introduce a reporter gene to a specific locus to be able to see that, something that utilizes GFP or selection.
And I have one more question.
Mm-hmm.
Can you really expect human cells to survive in a mouse environment?
Well, other studies have shown that you can. There's obviously some issues to be addressed, but the fact that these mice are immune suppressed so that they can, they don't get attacked by the mouse's native immune system helps. And it has been shown, like I said before, that even other groups have been able to transplant very similar cells to these into mice and allow them to continue growing.
What systems?
Huh?
What systems?
Well, they're actually, according to ViaCyte, they can transplant embryonic stem cells into the mouse and be able to continue generating insulin-producing cells.
As mentioned before, Mira came to this project. The strength of this lab before stem cell became available was our knowledge of development of human fetal pancreas. So we took pancreas from abortion clinics and different gestational ages, and we thought we performed maybe close to 1,000 transplants, manipulating the cells and look like islets, symbolic or something underneath the capsule, spring pancreas. So we developed quite a good reputation for doing this type of work. And we really show that when you take fetal cells from human fetal pancreas as early as 10-week gestation, the animal is able to absolutely differentiate them without teratoma. We can reverse diabetes that we induce chemically. So it's a very beautiful and reproducible system. Like, this is one of those experiments that works 90% of the time.
Again, like very naive questions. In the beginning, you mentioned that this transplant technology potentially has potential to alleviate the problem that you have to suppress patients, patients to transplant cadavers. So if this technology works, you can take patients' skin cells and redifferentiate them into beta cells, and then you'll be able to transplant them back to kidney or into the pancreas. Is there an issue of the immune attack of the patient again, that all that the patient have to be also suppressed, which is
Right. Well, the idea behind it and the hope is that we'll be able to have a point where they won't have to be immune suppressed since it is from their cells. However, there have been recent studies where
initial program diabetes mellitus.
Oh, you mean
Type
so
1.
autoimmune
Is that-
attack?
Exactly.
Okay.
Or does it happen only at a certain stage?
No, that would be a major concern. There's a couple of different ways that we could address it by either encapsulating the cells in something that's not permeable to immune cells. I know that Viasat's also looking at that. There's also, I mean, it would have to be a collaboration between immunologists and stem cell biologists to find a way to prevent the immune system from just destroying the beta cells again. Yeah, it is a concern.
It's our biggest problem right now is that we don't have anyone actively looking at this issue from the immune point of view. And diabetes is the 50% immunology, 50% is diabetes.
Any other questions from the committee? Okay, so general questions. Cece?
I have a couple. First, and these are all related to the transplants of teratomas, so I'll take your brain before you sleep. 50% of the mice develop teratomas.
Yeah.
What happened to the other 50%?
Oh, yeah, I
Did
didn't
the
really mention that
transplants?
today. So the other 50% didn't grow teratomas, but they also didn't generate any C-peptide, which just indicates that possibly all of the cells just didn't survive transplant.
So this then brings me back to the transplant and the data that you showed that you had very significantly reduced OCT4 and non-OCT levels in the cells that you transplanted, yet you still got teratoma. So the question is, and this is one that I don't know the answer to, but I want you to kind of think about and just kind of guess. That is, are these cells, is there a, when you transplant, say, 1 million or however many cells, this 1%, is there a selective growth advantage of the cells that are pluripotent, or is there dedifferentiation of the cells, or are you getting massive apoptosis of the cells? What's happening that's giving this selective advantage for the teratoma growth?
Well, I mean, that's the question.
Well, what do you think?
I think that actually the idea that there's a selective advantage for their growth makes the most sense to me, especially because, for instance, some reprogramming factors like c-Myc are cancerous transcription factors. That's a known oncogene. And so it's very possible that those pluripotent cells are just more hardy than the pancreatic progenitors that we're transplanting, and that once they are in that new environment, the other cells can't survive as well as they can. So that would probably be my opinion on what's going on. But, like, I mean, that's the million-dollar question.
You seem like that we should have done the transplant in the pancreas. following Dr. Cruz's question, that maybe there were some factors there that which inhibit teratoma and fate or basically development.
I think that would be really interesting to do. I mean, I definitely think that there might be ways to encourage that pancreatic development that we haven't found yet. You know, that maybe just transplanting the cells underneath the kidney is not enough to give them what they need to continue surviving. I mean, especially with the results where we saw no C-peptide, and we ended up with half of the transplanted mice, half of the mice that received the transplants not having anything at all. That does indicate that the cells aren't surviving for some reason or another. And we know that when we transplant them, they are expressing pancreatic transcription factors, and they do seem to have begun to take on a pancreatic sulfate. So something is going wrong with that one. And my dad has a question.
Well, it's along those same lines. You develop these cells to stage 5. How close are they to beta cells at that point?
They're still expressing, so we think that they're slightly immature, but close to beta cells at that point. They've been shown to express some of the different transcription factors for beta cells along with insulin,
Right.
including PDX1 in some of the work that I've done. However, they also are polyhormonal in some cases, so they express insulin, glucagon, and somatostatin, and that's indicative of an immature cell at that point. So they're close, but they're not quite the...
And is there a way to push them even closer to beta cells before you transplant?
There probably is, but so far I haven't seen any studies where people have been able to. The later stages of differentiation, there hasn't been as much of an. People haven't looked as much at signaling molecules and finding ways to fully optimize those to push them more towards beta cells rather than other cell types. So not right now.
I was wondering if the formation of teratomas is something like specific, a specific problem for the induced pluripotent stem cells versus these, embryonic stem cells.
Right. No, when we transplanted embryonic stem cells under the same conditions, we get teratomas with those as well. So it seems to be more of an issue of finding a way to weed out the pluripotent cells before transplantation.
The best test that we have of pluripotency is the formation of teratomas. So every time we receive the fighting cells, we transplant some seed, we will be initiated and we pay for that.
Of all the teratoma that you got, does any of them that is a pancreatic tumor?
No.
Or close to a pancreatic tumor?
No, I think that there was, in one of the histology samples, there was some tissue of pancreas, but not pancreatic tumors. No.
So another question is that when you do the implantation, so basically you differentiate the cell into endocrine beta cell, right? So it's not...
Well, when we transplant them, they're not at the endocrine beta cell
But
level
my
yet.
point is that it's only beta cell, right? But actually in pancreas you have axencrine cells.
Right.
Do you need to mix them up to give them like more amiable environment?
Well, that kind of goes along, I guess, with the idea of possibly transplanting them into the pancreas instead of underneath the kidney capsule, because then they would have access to those other pancreatic cell types. So it would be an interesting thing to try.
But actually in human, basically, they already have carried this kind of experiment in human, right? This beta cell transplantation.
You mean using
years
islets to...
Yeah, years ago, right?
Mhm.
So where do they transplant?
That goes directly into the hepatic vein, and so that's near the liver, basically. And so it's, again, that is not directly into the pancreas. And those cells are able to survive. But those are an actual beta cell. They're not a cell that we've differentiated and is close to a beta cell. It's an actual beta cell that's been functioning in a person just fine. So that's what we're shooting for, being able to make it. Yes.
So at the end of the differentiation protocol, what are the markers that you use to sort the cells that have to be
Transplanted? Well, we actually transplant them after stage 3, and what we have been looking at is their expression of NGN3, their expression of PDX1. And I mean, those are the main two, because PDX1 expressing cells are pancreatic progenitors. However, we can't really select for those cells yet. We're working on ways to do that.
Expressing a what? Messenger RNA or you?
Yes, messenger RNA, and with staining, you can see that they're expressing at the protein level.
So not 100% of the transplanted cells
Right.
were expressing.
Yeah, we know that.
There were some that were not expressing it.
Right. Yeah, and we know that that's the case. However, we just don't know
Silently
what it
expressing the
affects these.
I may have missed it, sorry Megan, but what was more efficient, the human embryonic stem cells or the iPS, as far as in vitro differentiation?
Human embryonic is still slightly more efficient. However, with just optimizing timing, we were able to see that the iPS cells could be better differentiated.
So as far as an exact number, you said slightly?
Usually embryonic stem cells, like with our addition of PB2, we were able to get up to almost 30%. Actually, it was lower than that for NGN3. Let me go back to that.
It's 27.
Okay. So for NGN3 cells at the end of stage 3, we get 12%. And for insulin-producing cells at the end of stage 5, these are the best conditions that we've been able to get so far. We get 30% C-peptide positive.
That's an iPS cells.
No, this is an embryonic stem cells. So that's why I'm saying embryonic stem cells are still more efficient than iPS. There's, my guess is it has something to do with just the differences in where the cells come from. There's probably some epigenetic factors there that are causing the iPS cells to not be able to be differentiated quite as well as ES.
So Ron, what percentage of cells are beta cells in the eye?
In the islet?
Yeah.
In the human islet? 50,
It was
60?
one of our experiments, so you better come with the answer.
I've already done my thesis over.
No, I like this much better. Turn your question.
It's more like 30.
Yeah, like 30%. So
Yes?
Yeah, after you wait for differentiation, do they respond to glucose stimulation?
iPS cells or the ES cells?
either one.
Either one. Okay. The ES cells, they do, but it's unregulated, and that's under the best of circumstances. iPS cells, I don't know if we've done glucose stimulation in iPS. We did? Okay. I didn't include it in my thesis work, so I don't know. But okay, yeah. So they do secrete insulin, however, it's not regulated by any means.
So respond to glucose stimulation is the main advantage for this kind of treatment. Am I right?
Yeah. I mean, that's the whole idea behind it, is that eventually we'll be able to differentiate cells that can respond to glucose just the same way that a beta cell would.
And that's a really important point, because when we were working with human phenotype pancreases, and we had these very beautiful results of being able to increase the number of beta cells in these undifferentiated cells, let's say from 10% to 80%. Still, in vitro, we could not get it. Very, very small. The only way was doing the transplant, waiting for 3 or 4 months, and then the cell was able to mature. As of today, nobody has been able to find which ones are the in vivo messages that make the cells responsible for glucose.
Any other questions?
Okay.
Thank you.
Thank you very much.